Welcome again to our new channel. Today we're going to discuss Huntington's disease. So let's get started. Huntington's disease is a rare disease that results in progressive degeneration of neurons in the brain. The degeneration is most pronounced in the striatum. If you remember, the striatum was composed of the caudate nucleus and the putamen. This is the major part of the brain that gets affected in Huntington's disease. But that's just early on. Later, other areas of the brain will be affected. The disease is most commonly inherited as an autosomal dominant manner, which means you just need to inherit one abnormal copy of one parent in order to get the disease. So let's say you have a gene A capital, A small, and this is the father. And you have A capital, and this is the mother. For any kind of gene, you inherit one copy from the father and one copy from the mother. So let's say this small a is the abnormal gene. So if you inherit, for example, the A capital from the father and another A capital from the mother, you're okay, you're normal. But if you inherit this small a from the father and another A capital from the mother, you get the disease. So you just need one abnormal copy of the gene in order to get the disease. This is called an autosomal dominant disease. This also implies that you have a 50% chance of getting the disease passed from an affected parent. So just as the same example here, you have a 50% chance of getting the small a and you have a 50% chance of getting the big A. So you have a 50% chance of getting the disease if you have an affected parent. There is also a 10% chance that you can just get the disease from a spontaneous happening mutation. So in that case, you don't need to have an abnormal father or mother in order to get the disease. You just get it spontaneously from a spontaneously happening mutation. Also, you need to know that this disease has 100% penetrance. And that means if you have the abnormal gene, you have 100% chance of displaying clinical manifestations. Having the abnormal gene is called genotype. And displaying the clinical manifestations is called phenotype. So when you are displaying the genotype of Huntington's disease, you have 100% chance of getting or displaying the clinical manifestations, which means the phenotype. This disease usually results in progressive movement disorders, cognitive disorders, most commonly dementia, and psychiatric disorders, most commonly depression. The disease onset is usually between the 30s and 40s. It can occur earlier in a process called anticipation, and we're going to talk about this in a minute. It results from a trinucleotide repeat expansion. Trinucleotide means 3 nucleotides, which are the cytosine, adenine, guanine, CAG. This expansion happens in the gene that codes for a protein called Huntingtin protein in chromosome number 4. How are we going to memorize this? Huntingtin, hunt, somebody is hunting. Hunt is 4 letters, so that means it's chromosome number 4. This is how you're going to memorize that Huntington's disease is an abnormality in chromosome number 4. This CAG repeat is encoding for an amino acid called glutamine. So this will result in polyglutamine expansion of the gene. This polyglutamine expansion will result in the destruction or abnormalities in the gene. The length of the repeat will determine the severity of the disease and will also determine the onset of the disease. So this has a link with this same process of anticipation. The diagnosis is mainly medical. It can be confirmed with genetic testing. Unfortunately, there is no cure for Huntington's disease. Now let's talk about the pathophysiology of Huntington's disease. And I would like you to please go watch the video of the basal ganglia in order to understand the anatomy and the physiology of the basal ganglia. The most vulnerable neurons to damage in the striatum are the ones that's projecting to the globus pallidus externus. So it's not all the neurons in the striatum gets damaged, it's most specifically the neurons that's projecting into the globus pallidus externus. And this leads to the abnormal movements. The striatum is not only involved in the control of voluntary movements, it's also involved in cognitive functions and behavior. And this explains why in Huntington's disease there's a variety of clinical abnormalities. Just as a quick review of the indirect pathway, here is the striatum. In the indirect pathway, the motor cortex receives impulses from the prefrontal cortex telling the motor cortex we need to stop an action. Then the motor cortex projects to a certain part of the striatum, not the entire striatum, a certain part of the striatum, telling the striatum we need to start the indirect pathway. At the same time, the common starting point for both pathways, the pars compacta, is sending D2 receptors into the striatum, also telling the striatum we need to start the indirect pathway. In that case, the striatum is sending GABA inhibitory impulses into the globus pallidus externus. This part over here is the defective part in Huntington's disease. So when you lose this inhibition of the globus pallidus externus, this will result in continuous inhibition of the subthalamic nucleus. So the subthalamic nucleus is now no longer working or is becoming continuously inhibited. So it's not going to work anymore on the globus pallidus internus. So now the globus pallidus internus is not stimulated to inhibit the thalamus. So now the thalamus is more active than before. And this will result in more impulses reaching the motor cortex. And this explains the abnormal movements. So generally speaking, the indirect pathway is less active in Huntington's disease, and the direct pathway in turn is more active. And if you remember from the basal ganglia video, we said when the dopamine level is higher, this favors more the direct pathway. And when the dopamine level is low, this favors more the indirect pathway. Another point we need to keep in mind is that dopamine and acetylcholine in the basal ganglia act like enemies. So when the dopamine level is high, the acetylcholine level is relatively low, and vice versa. So generally speaking, we can conclude that in Huntington's disease, there is generally decreased GABA, as we noticed here, and there is also decrease in acetylcholine, and this is because there is a relative increase in dopamine. Huntington's disease will eventually affect the entire brain, but most prominent early changes happens in the striatum. Later on, areas like the cortex, the cerebellum, the hippocampus, the thalamus, and the substantia nigra will all be affected. Atrophy of the caudate nucleus will result in characteristic secondary enlargement of the lateral ventricles. This is called hydrocephalus ex vacuo. Please refer to the video about Alzheimer's disease for more explanation of this point. The caudate nucleus forms the lateral borders of the lateral ventricles. So you can imagine if the caudate nucleus becomes atrophied, the ventricles will secondary enlarge. This is exactly what happens in Huntington's disease. So the atrophy of the caudate nucleus will result in the enlargement and the widening of the lateral ventricles. Not because there is a lot of cerebrospinal fluid, but just because more space is created in the ventricles by the atrophy of the caudate nucleus. Huntingtin protein is expressed in all cells, with its highest concentration in brain cells. The function of Huntingtin protein is still unclear. This protein contains normally between 6 and 35 glutamine residues. This is normal in normal individuals. But in individuals with Huntington's disease, they have more than 36 glutamine residues. The mutant protein contains the polyglutamine expansion, which makes the protein more prone to degradation into smaller fragments that accumulates within neurons. This accumulation will result eventually in the impairment of several cellular functions. The mutant Huntingtin protein can cause cell death by several mechanisms. Most importantly is mitochondrial dysfunction. The mitochondria has two important functions. First is energy production through ATP. This ATP is used by the cell to pump out excess ions and toxic substances out of the cell. These substances will accumulate within the cell without the presence of ATP, and this will result in cell death. The second important function of the mitochondria is the removal or the fighting of the oxidative stress through the production of reducing agents, so this will prevent the accumulation of reactive oxygen species. The accumulation of these reactive oxygen species can also cause cell damage. Another important mechanism that may be included in the pathophysiology of Huntington's disease is the excess glutamine. The excess glutamine can be toxic to the neurons and can cause neuronal death. As we said earlier, Huntingtin gene is located in chromosome number 4. Hunt is 4 letters. This makes you remember that Huntington's gene is located in chromosome number 4. As we discussed earlier, the pathology of Huntington's disease includes a trinucleotide repeat expansion. This trinucleotide repeat expansion happens within the coding region of the chromosome. So the trinucleotide repeat expansion in some diseases can happen within the non-coding region, and this can result in decrease or increase in the expression of a certain gene. In other diseases, which includes Huntington's disease, the trinucleotide repeat expansion happens within the coding region of the protein, and this can lead to structural changes in the protein, which would result in diseases. The triplet expansion is caused by a phenomenon called DNA slippage, or slipping of the DNA that happens during DNA replication. As we all know, the DNA replication is going from the 5' to the 3' direction. And that's in the daughter strand, because the template strand is being replicated from the 3' to the 5' direction. So basically what happens, the template strand is being replicated in the 5' to 3' direction by DNA polymerase. When the DNA polymerase encounters an area of triplet repeat, it will stop. It will temporarily stop. And this will result in the partial detachment of the daughter strand. Then the daughter strand will reattach to the template strand, but to an area that's upstream or earlier in the sequence of the template strand. And then the DNA polymerase will reattach again and then will repeat that segment again. This is all because all these triplets here are the same. So the daughter strand will reattach because they all look alike. When the daughter strand attaches to the template strand to an earlier or upstream segment, this will result in the creation of a loop. This makes it look like the daughter strand has slipped, and this is where the name came from, the DNA slippage. Because it sounds like the daughter strand has slipped. This slippage over generations will expand, and this will result in the expansion of this trinucleotide repeat over generations. This will result in a phenomenon called anticipation. So the expansion increases with subsequent generations, and this will result in anticipation. The anticipation means the signs and symptoms of the genetic condition tends to become more severe or even earlier in onset by the time or with generations, or from one generation to the next. Also, this disease has 100% penetrance, which means all people with the genetic mutation will develop the disease. In other words, all people with the genotype of the mutation will develop the phenotype of the disease. The signs and symptoms of Huntington's disease includes motor dysfunction, cognitive dysfunction, behavioral and psychological dysfunction. And this is all because of the dysfunction of the striatum. Motor dysfunctions includes chorea, which means brief, irregular involuntary movements that appears to flow from one muscle to another. Athetosis is a slow involuntary writhing or twisting movements of the hands, fingers, or toes, and also rigidity. The cognitive disorders include amnesia, lack of concentration, and dementia. The behavioral and psychological includes aggression, irritability, anxiety, and depression. So, whenever you encounter a middle-aged patient who displays abnormal movements, in addition to some memory disorders, plus some behavioral abnormalities like irritability or anxiety or aggression, you should think of Huntington's disease, especially with the presence of family history. But as we discussed earlier, this might not be the case because some cases of Huntington's disease are just sporadic mutations. The length of the repeat will determine the severity and the onset of the symptoms. And we discussed that and we call that anticipation. Life expectancy is about 20 years from the onset of the symptoms. Aspiration pneumonia is a common cause of death. The diagnosis is based on the clinical manifestations primarily and the family history, if it's present. It can be confirmed with genetic testing, which will demonstrate the trinucleotide repeat expansion in chromosome number 4 on the Huntingtin gene that encodes for the Huntingtin protein. Unfortunately, there is no cure for Huntington's disease. Now let's talk about an important topic that's highly relevant for the boards. It's called histone deacetylation. First, let's talk about histones. Histones are highly basic proteins that are found in the eukaryotic cell's nuclei. They help to package the DNA into structures called nucleosomes. The histones contains large amounts of amino acids, lysine and arginine. The length of the DNA within the cell is too long. So in order to package this DNA into smaller structures, we have these histone proteins. So the DNA binds or winds around these histone proteins, and this will decrease the size of the DNA strands. And the reason why the DNA binds tightly to the histone proteins because the negative charge created by the phosphate groups of the DNA interacts with the positive charge of the histone proteins. In addition to help package the DNA into smaller structures, this tight binding between histone proteins and DNA has another function, which is the control of gene expression. So when the binding is tight between the DNA and histone, the gene expression is decreased. When this binding is more loose, the gene expression will increase. Generally speaking, the nucleosome is made up of 4 types of histone proteins. These are called H2A, H2B, H3, and H4. Each one of them has 2 copies, so we end up with 8 histone proteins within the nucleosome. This is called octamer, where the DNA, as we said, wind around these histone proteins. There is another type of histone protein called H1, but this is called linker histone because it binds with the linker DNA. Histone proteins contains large amounts of lysine amino acids, and these lysine amino acids are positively charged because they contain an N-terminal with a positive charge. When this N-terminal becomes acetylated, or in other words, an acetyl group is added to this N-terminal, it loses the positive charge as we see here. So it becomes no longer positively charged, so it will not interact with the phosphate groups which are negatively charged within the DNA. So this will free up the DNA. So this will increase the gene expression. On the other hand, or in the other direction, histone deacetylases removes this acetyl group of the N-terminal of the lysine residues. So this will restore the positive charge that will interact with the negatively charged phosphate groups of the DNA. So this will lead to more tight binding between the histone and the DNA. So this will decrease the gene expression. What's the bottom line of all that, or why we are discussing all that? Because one of the treatment strategies of Huntington's disease is histone deacetylase inhibitors. So these will help to increase the acetylated histone, so it increases gene expression in Huntington's disease patients. And recent studies have found that this strategy helps these patients. So histone deacetylation may play a role in the pathogenesis of Huntington's disease. And this is the bottom line of all this histone acetylation and deacetylation related to Huntington's disease. Alright, so this is the end of our video. Thank you very much for watching. Please don't forget to subscribe and see you next time.